Inside the cranium, the most visible part of the brain is the cortex, considered the seat of intelligence. Buried under its folds is the limbic brain, which governs our emotions and motivations. This is where drugs go to work. All drugs work by hijacking the brain's communication system. Our brain, indeed, spends most of its time chatting to itself. In order to keep things running, it relies on a network of ongoing conversations between our 100 billion nerve cells, or neurons. The neurons exchange information by sending each other chemical messengers called neurotransmitters. Whether it has to do with pain, pleasure, or the rate of information exchange, the neurotransmitters act as regulators, maintaining a complex and fine-tuned equilibrium which is essential to all of our bodily functions. Between the neurons are little spaces which are always busy with activity. It is here, in the synapse, that the neurotransmitters do their work. They transfer information from one neuron to the next by binding themselves to the appropriate receptors. These systems exist in human beings and in all mammals, including the smallest of them all. Studying how drugs work would not have been possible without rats or mice. They share many of the same genes as human beings, and drugs stir up the same kinds of tempests in their brains. In fact, all of humanity is morally indebted to these little creatures. We can intoxicate rats, manipulate their genes, even cut them up into little pieces. This is the terrible but necessary task of laboratories that compare the reactions of normal or control rats to so-called knockout rats. This technique consists of knocking out certain genes in normal mice, for example, the gene that controls the production of a certain neurotransmitter. These genetically modified mice are called knockout mice. Dozens of neurotransmitters have now been identified, but the one that plays a key role in feelings of pleasure and the desire for drugs is the aptly named dopamine. This is what dopamine looks like under a microscope, in its crystal form and its chemical structure. Bruno Giraud studies the wide-ranging effects of this tiny molecule in the brains of normal and knockout mice.
Toutes les drogues qui sont utilisées chez
All drugs taken by human beings, nicotine, alcohol, opiates, cannabinoids, psychostimulants, have one thing in common, which is that they stimulate the release of dopamine. So dopamine is at the core of the brain's reward circuits, as well as, of course, its pleasure circuits. Dopamine is also released by compulsive gaming, sexual activity, and eating, for example. In fact, sexual activity and eating are very important for the production of dopamine. In other words, they are survival behaviors, necessary for the survival of the species. What drugs do is imitate these activities. They hijack the system, they take advantage of it. The brain wasn't originally made to be dependent on drugs. It was made to manage the reinforcing systems that lead us to engage in sex and eating. Drugs work by deceiving and taking over the system.
This experiment reveals one of the functions of dopamine. The box on the right contains a normal mouse. On the left, a knockout mouse, which lacks a key element in its dopamine circuit, the transporter. Unlike the normal mouse, the knockout mouse seems ready to jump into the ring. If cocaine is injected into the normal mouse, it starts to behave in exactly the same way.
Here you have the nerve ending of a normal animal, which releases dopamine. The dopamine will then bind to receptors on the receptor cell here. And quite quickly, the dopamine is recaptured by the dopamine transporter and taken back into the neuron. In the normal mouse that has been injected with cocaine, the cocaine, which, like amphetamines, is a psychostimulant, will block the dopamine transporter. In the knockout animal, you have everything: the nerve ending, dopamine release, activation of receptors, except the reuptake. So a lot of dopamine remains here in the synapse, and it overstimulates the receptors, which is why you see this hyperactivity. These mice are much more active. Their behavior is in fact identical to that of the normal mouse you saw who was injected with cocaine. Once we've understood how the system works, we can identify which levers to push in order to increase or decrease the amount of dopamine that is released. Afterwards, we can test our hypotheses on human subjects using the pharmaceuticals that we have or those that will be developed in the next few years. Obviously, with this animal model, we can't ask the mice about their mental disturbances, which is what we want to explore in human beings. But everything that's behind these mental disturbances, the neurobiology, is exactly the same. It's just that the neurobiology is expressed differently. In mice, for example, through movement, and in human beings through mental confusion.
When stimulated by cocaine, dopamine doesn't just go anywhere in the brain. It concentrates in the limbic brain, around the striatum, and especially in the nucleus accumbens. Studies of this tiny but crucial area over the last 10 years have created a small revolution in the neurosciences. The nucleus accumbens is generally thought to be the pleasure center, which is stimulated by dopamine.